All right. I think it's all right. It's recording. So, continuing on, we are very honored to have our speaker today. She is a postdoctoral researcher in the Center for Translational Cancer Research at the Brown Foundation and Institute of Molecular Medicine. Her talk today will be about the development and the application of various peptide conjugates that target SSTR2 for the detection and therapy purposes in neuroendocrine tumors. So without further ado, Dr. Solmaz, I'll let you continue.
Okay, thank you so much for the kind introduction, Rachel. Can I share my screen?
Yes.
Yes. Okay. Can you see my screen fine?
Yes, it is in like the PowerPoint version, I think.
Okay.
Yeah, all right, there you go.
Yeah, perfect. All right. Let me set this. All right. Thank you, everyone. Good evening, everybody in the UK. Good afternoon, everyone in the US. So thanks for being here. And today I'll be talking about novel applications of SSTR2 targeting peptides in neuroendocrine tumors. But before I start the presentation, I want to thank the organizers for inviting me and giving me the opportunity and the time to present this work. I'm so excited to share this work with all of you. I have nothing to disclose. And I actually divided this presentation into two different sections. In part one, I'll be talking about the bioconjugate, the somatostatin-based bioconjugate that has been developed in our lab that mostly target localized neuroendocrine tumor. And in the second part, I'll be introducing a PDC for metastatic disease. So we know that surgery can be a curative option for neuroendocrine tumors, especially for localized disease, if or when all the cancerous tissue is removed. However, like any other cancer surgery, this can be challenging, finding the tumor, all the lesions, especially when we have multifocal disease or we have these small hidden lesions. So with the current standard of care surgery technique, we have 15 to 45% of surgical cases with incomplete resections, which actually causes poor prognosis because they're associated with tumor recurrence. They can be associated with tumor recurrence and other complications for the patient. So because of this critical need for accurate tumor detection during oncologic surgery, new approaches, new technologies have been developed and introduced to the ORs. One of them is fluorescent-guided surgery. Fluorescent-guided surgery, as you may gather from its name, it actually uses a fluorescent agent to enhance surgeon ability to locate, to find tumor in a more precise way. And the goal of this technology is to improve surgery outcome with actually helping the surgeon to remove as much as tumor as possible while sparing the normal tissues. So there are several tumor-targeted fluorescent-guided surgery agents in development in preclinical studies, but currently one has been approved by FDA for ovarian cancer surgery not long ago. There is none currently for neuroendocrine tumor surgery, and so our group thought that definitely this patient can benefit of an agent with the same that has actually this that can help the surgeon in intraoperatively. That's why we developed the FGS agent that I'll be talking about. The first question when we decided to develop this agent was how we can develop a target agent for FGS in NETs. We know that majority of patients with neuroendocrine tumors, they overexpress SSTR2, somatostatin type 2 receptor. It's a receptor, it's a protein molecule that is present on the cell membrane of these tumor cells and can be a great target for imaging or therapeutic purposes. On the other hand, we have somatostatin analogs that are synthetic analogs of the natural ligand for this receptor. And these somatostatin analogs, they have been in the clinic for over more than 4 decades now, and they have shown excellent binding properties towards the receptor. So they bind to the receptor in a very specific manner with high affinity, make them ideal delivery vehicle to deliver imaging or therapeutic agents, as we see in the development of, for example, imaging agent, gallium dotatate, and in the development of therapeutic agent, lutetium dotatate. So they've been used to deliver imaging or therapeutic isotope, radioisotope to the tumor. So long story short, we have this great tool of receptor ligand in neuroendocrine tumor that we can pretty much use it to deliver anything in a specific manner to the tumor. So we use the same concept, and we selected gallium dotatate, the PET radio tracer gallium dotatate that is being used in the clinic for detection, surgical planning, and follow-up in patients with neuroendocrine tumors. So this was our model. We converted the PET radio tracer into a fluorescent counterpart, as you can see in the bottom. So our chemist, Dr. Ghosh, actually made this some modification in the gallium dotatate to be able to add a fluorescent dye to make this FGS agent. If we compare these two structures together, we see that the talk part, the binding part in blue, is intact. We didn't touch that part because the binding properties of the agent is the reason we selected the agent, right? So we did not change that part. What we changed was the DOTA, the chelator. Chelator is that part of the molecule that holds the isotope in place. So we modified the chelator. And if you compare these two structures, they're very similar, except for the new chelator that we called it MMC, multimodality chelator, has an extra arm that enables us to add a fluorescent agent. So this is the final agent that was synthesized, FGS agent, and we called it MMC FNAR tactile. And after synthesis and purification steps, we were ready to test the agent in the cell studies, right? That's what we do in the lab. So we have done several in vitro studies, cell studies, but because of the time, I'm only showing one of them, confocal microscopy here. We selected a cell line that has SSTR2 expression. So this way we can evaluate the binding of the agent to SSTR2 positive cells. As you can see, after adding the agent, we can clearly see the signal that comes from the near-infrared dye. So it shows that agent bind to the receptor positive cells. To evaluate a specificity, we perform a blocking study. On the right, you see the same cells, but before adding the FGS agent, we actually blocked the receptors, saturated the receptor with excess amount of nucleotide, and that reduced the fluorescent signal to the minimum. So it further confirms specificity of our agent. After completing all the in vitro studies, we were ready to go and proceeded to in vivo studies. We had several animal models. I'm showing one of them. We developed these bilaterally implanted xenografts that in one shoulder they had SSTR2 positive tumors, and in the opposite shoulder they had receptor negative tumors. This way we could evaluate the specificity of the agent in vivo. And as you can see in this video here, after injecting the agent into the animals, we were clearly able to see accumulation of the signal in receptor positive tumor, whereas there is minimum signal here in receptor negative. So the agent binds specifically to the tumor, to receptor positive tumors in vivo. I also want to draw your attention to the background signal here. You see the background is relatively clean. And this is one of the powerful part of our drug design, because we actually use gallium that has a beautiful PK in the clinic, binding to the tumor, low non-specific binding and clearance through the kidney. When we added the near-infrared dye, even though the physicochemical properties of the dye changes the final everything in the molecule, we still have this beautiful clearance through the kidney. You see the signal intensity in the kidneys, and there is not much background signal in non-specific, in non-tumor tissues. So here, we actually simulated what's a clinical workflow to use with our agent in the clinic, right? This is a fluorescent agent that targets SSTR2. For a patient to be able to receive this agent, their tumor should express SSTR2. So the patient, the proposed clinical workflow is for the patient to undergo a gallium DOTA scan to find out if the patient is SSTR2 positive, and if they're receptor positive, if their tumor is resectable, then they are a candidate to get the FGS agent. So we simulated this in an orthotopic model of the disease. We implanted born SSTR2 cells into the pancreas of the mice. After the tumor were formed, actually we could not see visually that the tumor is growing. We didn't have tools to understand if the tumors are growing or not, unless you see the changes in the behavior of the mice that are like showing some symptoms that are associated with having a tumor. So after the tumor growth, we injected the mice with the radio tracer gallium DOTA, and we performed a PET-CT study. As you can see, PET-CT showed the tumor local, actually, with the PET-CT, we found the location of the tumor here. This is the kidney and this is the bladder, the clearance of the gallium DOTA. And then 2-3 days later, we injected the same mice with our FGS agent. As you can see, after fluorescent imaging, we could see the fluorescent in the same location that was detected with the PET radio tracer. So we removed the tumor, we imaged the wound bed, there is no signal here. We evaluated the harvested organs ex vivo, and everything ex vivo was confirming our observation in vivo. We have accumulation of signal here, background tissues are clean, and we notice a little signal in the spleen. When we performed histology, we confirmed that the metastasis probably originated from this tumor, and our agent could pick it up, which was very interesting. And infrared scan of the sections from the tissues, wherever we have IHC positive areas, the pattern of the signal was very compatible to the IHC positive areas. So that was successful. So far, we showed that our agent binds specifically to the tumor in the conventional tumor models, and we can actually detect the tumor with the fluorescent signal. Next step, we wanted to test the translational potential of our agent. So we selected this PDX model that we were very lucky that Dr. Ear, actually, she developed this model, and we know how challenging it is to develop a PDX model of neuroendocrine tumors. So we applied our agent in the model that she developed. We had this PDX model. These are patient-derived xenograft. It means that the tumor comes from the cells that are extracted from actual patient tumors. So these are different biology and everything than the regular cell culture when we implant them. The tumor, when the tumor is formed, is more close to the actual human tumors. So we have this receptor negative PDX and receptor positive PDX. The mice were bilaterally implanted again. And after injection of our agent, As you can see, again, we were able to show specificity beautifully, and the clearance again through the kidneys. Ex vivo confirms in vivo observations, and the gold standard histology downstream with H&E and IHC staining for SSTR2. We confirmed presence of the receptor in receptor positive tumors and the absence of the receptor in the receptor negative tumors. And as you can see here, the fluorescence signal also correlates with the distribution of IHC positive area. One step further toward testing the translational potential of our agent, we tested binding of the agent in the actual human tumor tissues. So we always know that there is a gap between preclinical development of an agent and true clinical application of an agent. We wanted to bridge this gap. That's why we collaborated with NET surgery group here at MD Anderson. We received these tumor tissues, sectioned them, stained them with H&E and IHC for SSTR2. We also stained these sections with our FGS agent. As you can see, again, we have excellent co-localization of the fluorescence signal with wherever we have IHC positive tumor. So overall, this agent showed promising results in preclinical studies, and currently we are completing the steps that are required for the first in human study, hopefully in 2024 here at MD Anderson. So moving on to the second part of the presentation, I hope that I haven't overwhelmed you yet, but I'm going to introduce this part is going to be faster. We actually developed, with the success we had in developing a fluorescent agent using this MMC technology, that actually motivated us to develop a PDC. So we swapped this near-infrared dye for a cytotoxic drug. The payload that we selected was temozolomide. Temozolomide is an alkylating agent, and it's a standard of care for the treatment of neuroendocrine tumors with a response rate of 30 to 70% in patient with NETs, alone or in combination with capecitabine. It's actually a prodrug that in physiologic pH, it undergoes several spontaneous hydrolysis process, and it releases this highly reactive ion, methyldiazonium, that this ion actually attacks the DNA and methylates the DNA in different positions. One of them is O6 position of guanine, which is this adduct, this lesion is highly toxic. That's what actually causes that's the lesion that is responsible for most of the cytotoxic effect of temozolomide. On the other hand, in the cells, repair system of the cells, they have a direct repair system for specifically this adduct, which called MGMT, or methylguanine methyltransferase. And it's an enzyme. The role of this enzyme is to directly remove the O6 methylguanine adduct and repair the DNA. So there is a negative correlation between presence of the receptor and response to the temozolomide. So wherever we have higher MGMT expression, the response to temozolomide is lower, and vice versa. In pancreatic neuroendocrine tumors, we usually see better response with temozolomide because PNETs are usually in 25 to 50% are MGMT deficient, whereas in almost all carcinoid cases, we have intact MGMT that results in worse outcome with temozolomide treatment. One unique characteristic that this enzyme has is it can be overwhelmed, it can be exhausted, which means if actually after it performs its repair function, it undergoes proteasome degradation. So it gives us the opportunity to overwhelm this enzyme by delivering more drug and causing more damage. So when we concentrate the drug inside the tumor, we cause more damage. The enzyme is going to actively repair those damage. And on the other hand, it goes through the proteasome degradation. So we have a lower enzyme over the time after treatment that this way this defense shield is broken and we will have better outcome in treatment with temozolomide. This opportunity actually resulted in having dose-dense temozolomide regimen in glioblastoma, which temozolomide is the first-line treatment. However, with dose-dense temozolomide, the problem is substantial hematological toxicity. So our hypothesis was to use a targeted system to be able to concentrate the drug in a targeted manner in neuroendocrine tumors without harming the normal tissues. That's why we developed this PDC. Again, we used gallium dotatate as our target, as our model molecule, and we converted this. We use again MC technology here, and instead of near-infrared dye, we added the cytotoxic drug temozolomide here. The interesting thing in this drug design is the MMC part is still a chelator, so we can radio label this drug. This gives us tremendous opportunity for optimizing the drug in a non-invasive manner. In the next slide, I show you the application, how a radio labeled drug conjugate can help us optimizing a drug and actually non-invasively track the drug and by distribution of the drug in vivo. So again, the talk part, the binding part, we did not change this part, and what we changed was the chelator, and that's the drug that we added. We always in vitro studies, because we use this as a model, we always use gallium dotatate as a benchmark to evaluate the performance of our agent, mostly in vitro studies. So we started, let me add something here. So peptide conjugate. This peptide drug conjugate has two important arms. This arm is binding part, and this arm is cytotoxic part. After we synthesize the drug, we wanted to step by step evaluate to make sure this conjugation did not affect neither parts, neither arms. So we started to evaluate the binding properties of the peptide drug conjugate first. We use the radio labeling potential of the drug. We radio label the drug conjugate, and then we also radio labeled gallium dotatate. We evaluated the binding affinity of the drug versus dotatate, and as you can see here, it beautifully showed that almost there is no changes in the affinity of this new drug for the SSTR2 receptor compared to dotatate. We also perform a radioactive uptake study in different cell line with different expression levels of SSTR2, and we actually observed that binding of the agent was comparable, the amount of binding was comparable to the amount of binding of gallium dotatate in different cell lines, and the percentage of binding was correlated with the amount of SSTR2 receptor available. So we have higher receptor here, more binding, lower receptor in IMR32 and A69 cells, so lower binding. And there is no binding in wild type cells, ACT wild type cells, that they don't express any SSTR2. So this shows a specific binding of our molecule, and also in blocking studies, we were able to block the binding that further confirms a specificity. So this is a PDC. And we have temozolomide that should enter the cell, gets to the DNA to be able to cause the damage, right? So another thing that we wanted to make sure our agent, another properties we wanted to make sure that this drug conjugate has is to internalization, actually is to be able to internalize after binding. And we evaluated that, we using the radio-labeled compound. And as you can see here, after 1 hour of incubation, we found almost more than 60% of the bound fraction was internalized. And I just need to mention that we use talk that an agonist and agonist internalize after binding. So even the agonist properties of the molecule was retained, which was very interesting. So in the second part, so far, we confirmed that our drug is binding specifically to the receptor positive tumor. In the second part of the project, we wanted to make sure that the temozolomide also retain its effects, right? The cytotoxic effect and DNA damaging effect mostly. So we performed studies that they measure the DNA damaging effect of the drug, COMET assay and gamma-H2X assay. With COMET assay, we measure single and double strand DNA break. With gamma-H2X assay, we measure the double strand break. We had untreated cells. Actually, we use IMR32 cells here. They are receptor positive, and they have endogenous expression of SSTR2. We have untreated control. We use temozolomide as a positive control here, and we have our PDC. We also perform blocking studies to evaluate the specificity of our agent for the SSTR2 receptor. As you can see here, and the indication of a COMET positive is this tail that you can see in the TMZ and PDC, where untreated cell, the enclosed is intact, so there's no damage. In TMZ, we are able to see these tails that they show the damage caused by the temozolomide, by the drug. And in our PDC treated group, we also we were able to see the damage similar to what we saw in temozolomide group. And interestingly, in the blocked group, the damage was actually reduced because the receptors were blocked, were saturated, and the drug could not get inside the cell. The gamma-H2X assay also confirmed our observation with COMET assay. So it showed the receptor-mediated DNA damage caused by our PDC. Here we tested our central hypothesis that if we deliver more drug into the cells, we can overwhelm MGMT enzyme. So we had the cell line IMR32, which is MGMT positive and SSTR2 positive. And we treated IMR32 cells in different groups with free temozolomide, increasing concentration of our PDC, and a pseudo substrate for MGMT that is very efficiently deplete this enzyme called O6VG. And Western blot studies showed that when we have higher concentration of the drugs in all the groups, we had more MGMT depletion. But the MGMT depletion was obvious in all the tested groups. So we were able to confirm that our PDC actually reduced the MGMT content of the cell in a dose-dependent manner. However, we see a little bit like higher intensity of the band compared to free temozolomide, which was expected because free temozolomide diffuses to the cells, but here we have limitation of dependency to the receptors, so that was expected. And for the last part, actually, this is the main application of, this slide shows the main application of a radio-labeled PDC. Whatever we have shown in vitro was that our agent binds to the tumor and it can cause DNA damage. Next step was to evaluate these two in vivo, right? So we used the radio-labeled drug conjugate to track the drug in vivo in a non-invasive manner. So we radio-labeled the drug, we developed this xenografts that they have bilateral tumors, bilaterally implanted with receptor positive and receptor negative tumors, injected this xenograph with a radiolabeled PDC and performed a PET-CT scan followed by biodistribution study. And as you can see here, this is the result of our PET scan only 1 hour after injection of the drug. We see clear accumulation of our drug in receptor positive tumor, whereas there is nothing almost in receptor negative tumors. The clearance is through the kidneys and the bladder. The quantification also confirms this high accumulation of the drug in the tumor, clearance through the kidney, and very interestingly, very minimum drug accumulation in non-target tissues. So, and the last part of this presentation, I'm showing this the same thing that we have done in H69 xenograph, because this is a more relevant model in HCT that we have used. HCTs are transfected with SSTR2, so they overexpress SSTR2, but this is a cell line that expresses SSTR2, minimum level of SSTR2, and we were able to show that our agent was successfully bound to the to these low expressive tumors, and we had the same BioD and clearance of the drug in the same in H69 xenograph. So this is actually still an ongoing project. We are optimizing the drug conjugate with probably changing the payload to a more potent payload, but from the same class, because we have challenges to use temozolomide in vivo to be able to see the effect of the drug. We actually used this strategy as a proof of concept to show that our MMC technology allows us to develop a PDC that has this great pharmacokinetic, and if we switch the payload to a more potent payload, we hopefully see what we expect to see with a PDC. So I guess with this, this concludes the presentation today, and I'd be happy to take questions. Thank you for your time.
Thank you so much. That was an excellent presentation. Very exciting data in the field of SSTR2. I'm going to open the floor. Do we have any questions? I know we've got several SSTR2 experts on the call, so I'm excited about this discussion. Renata?
I have technical questions. So what is the cell line, HCT? This is neuroendocrine tumor cell line, or SSTR2 is overexpressed?
Yeah, these are colon cancer cell line, but they widely used for neuroendocrine tumor because it's an established transfected cell line that has SSTR2, and we had them in the lab, and we use them for this mostly for binding studies. That's why we always start our evaluation with this model, because it's like our standard model here. But we also had one SSTR2, which is more relevant to the disease, and we had H69 xenografts.
Yes, I think I know. I think I'm familiar with Dr. Azriani work. So now he was coming to us, I think, few times for meeting with our radiochemist Susie Lapi. So now I'm kind of recalling, this work.
Yeah, definitely.
Another question, I think I will have to Paul. So, remind me, your PDX model is neuroendocrine tumor, or this is FEO? What is the PDX?
Yeah, it's actually neuroendocrine neoplasm, like a neuroendocrine carcinoma that we characterize. It was one of them is actually from the ampullary tumor, and that one has it's really other than the fact that it has the P53 and RB mutation, it actually has like really high level of like SSTR2 and the other neuroendocrine tumor markers. So that's why we think it's a nice practical model for technology development, such as the amazing reagents that Solmaz presented to us today. But yeah, unfortunately, we're still working on the neuroendocrine tumors, and it did take like 6 months or 8 months to grow, a tiny little tumor that's really hard to propagate, and so yeah, it's a challenge.
So this is pancreatic neck
Yes.
neuroendocrine cancer. Okay,
Yes.
great, thank you. Yes, this is a great presentation.
Thank you.
We have more questions. I had a couple questions for our speaker. They're kind of more technical. The first question is the HCT 116, are those into nude mice or black 6 mice?
Yeah, nude
or
mice.
biopsy? Nude mice. Okay, so it's all right, it's a xenograft. And then my other question was, could you explain how you determined the percent internalization when you were doing the binding studies?
Yeah, of course. So after binding studies, we use this acid wash protocol. So we wash the cells with acidic solution. This way, whatever receptor we have on the cell membrane is actually detaching, or we're removing the receptor on the cell membrane. Whatever count or signal we get from radioactive comes from inside the cell. Was that clear?
Yes, thank you.
Yeah.
And then I had one more question I thought of. When you have the MMC, I believe, chelator, you had mentioned that in the future you'd be able to radio label the molecule. Will you still plan on using gallium, 68 gallium, to radio label that molecule?
It depends on the application. Well, there are several things you can do. When the chelator is there, you can potentially add the therapeutic radioisotope or imaging radioisotope. So it depends on what application and what is the purpose of a study. But yeah, definitely we can use imaging or therapeutic. It's probably, I mean, we always, in research, we are scientists, we're thinking about, oh, I want to combine this radio tracer with this PDC, but we don't know how it's going to affect the actual molecule. But you can add whatever radioisotope that you can actually feed into that chelator.
Makes sense. So you could, so potentially there could be in the future, you could add like lutetium there and have maybe a synergistic cytotoxic effect when you have that beta emitting radionuclide with the cytotoxic nuclide.
Exactly.
Ah, wonderful.
Yeah.
Wonderful. Do we have any other? What's that? Sorry.
Especially, we know that we have temozolomide is being used as a radiosensitizer in the field in combination with lutetium, right? With PRRT. So if we can use a more potent temozolomide, like any other drug in the same class that is more potent, and combine the whole molecule with lutetium, so I don't know, it can be another way of developing an agent with higher efficacy.
Yeah, that's very exciting. Very exciting.
Yeah.
That's neat. Maybe lower toxicity.
Lower toxicity.
I have a question. So yeah, so that was really interesting that you were, I had the same question as Rachel about how did you tell if it was internalized or not. And so as a follow-up to that, have you looked at, and maybe some others have looked at this, quantifying like maybe how many, like what the cell killing potential is. Like if you wanted to know how many molecules a cell had to uptake in order to die, it seems like that's the kind of thing that you would do, perhaps.
Yeah.
Is that going to work out there?
Yeah, we haven't looked into that, but that would be a very interesting question to answer. It's very interesting. But but to be honest, there is a problem with temozolomide. It's a tolerable drug. So to get to that number that actually can kill the cell, it's definitely needs more concentration of the drug, which I don't know if it's going to be possible with this targeted approach. That's why we're thinking about changing the payload to a more potent. But yeah, when we have that highly toxic molecule, that's a question that needs to be answered.
And in my case, it came up in the context of looking at wanting to develop a computational model for like lutetium therapy. So definitely got curious about like, well, how many counts does it take to kill a cell?
Exactly. Yeah.
And that's almost like, and I hadn't thought about it in terms of the drugs, but you could get some really interesting, if you will, micro PK/PD things going on if you can get a really accurate, precise count of how many molecules were absorbed. That's cool.
Yeah, definitely.
Cool. Awesome. Thanks.
I was going to pitch in, John. That's a great point, and Solmaz, I think the results looks like really encouraging, and I think maybe, although like it's hard to get like the cell death, because that when the cells is dead, and it's like it's hard to like actually physically track them. But I feel like your DNA damage agent and like experiments, either gamma H2AX or actually the Comet assay, and those, if I think, John, if you can use those as a proxy for like cell death, and then you can in theory, you should be able to kind of model that. And that's really nice because it gives you like a single cell, almost like a single cell resolution. And then you can actually do like modeling with your population effect and all that. So I feel like that could be a starting point. It's not definitely, it's not this ultimate cell death assay that you want, but it feels it's feasible, and you have these. And so I feel like it's a nice starting point there.
Yeah, definitely. Yeah, definitely. That can be. I don't know how exactly we can use this technology, this counts, because when we have this receptor internalization, the whole molecule undergoes this intercellular trafficking, right? Lysosome. Endosome, lysosome. So I don't know if the number of the molecule that gets inside the, let's say, nucleus and causes the DNA damage, we can have access to that. It's still bound to the radioisotope when it gets to the nucleus. So these are the questions that we have to answer. If the molecule gets chopped in another places, and it's simply, I don't know if the final molecule, the final effective molecule, has the radioisotope with it or not.
Well, if it's with a therapeutic radioisotope, then your counts will still be valid. Perhaps one could consider starting there. I know that's not the line of research that you are thinking about right now. So you raise a good point that in the context of wanting to look at this combination, the MMC combination that you have, then it might not, you'd have to be more thoughtful than would if it was simply a therapeutic ligand. That is to say, it's a therapeutic radionuclide. But yeah, that's neat. I'm still very curious also about, because if you can localize where the dose is going, then you can start to look at microdosimetry of the radiation. And I think that's a really interesting thing in terms of looking at the heterogeneity that is generated by maybe certain cell structures, and why do maybe ultimately leading to maybe trying to answer the question, why would some respond and some respond less well. Well, and it could have to do with that dose heterogeneity that is generated. So yeah, that's a really interesting topic. Happy to talk about that stuff more. Thanks.
These are great questions. Do we have more questions?
I do have a question, but I'm a little bit embarrassed to ask because I was a little late, and I'm so sorry because I needed to attend a mandatory meeting and finish later. But I'm looking forward to listen to your talk once it's available. But I do have a question just because I started watching about the DNA breaks with temozolomide, and just based on the discussion about that, you know, it seems like you will require a higher payload of temozolomide, if I understood well, in order to have some efficacy. That say, if you could prove that you have this enhancement of DNA breaks with temozolomide, how about the whole theory about DNA repair inhibition? So would you be willing to do a DNA repair inhibition to enhance efficacy yet less toxicity?
I don't think I got the question. So did you ask about using the DNA repair system as a target?
Inhibitors to enhance the DNA to because based on the graph that you show that temozolomide increased DNA repair, correct? I'm sorry, single-stranded breaks. My apologies, single-stranded breaks. That's what it does with temozolomide. So, and if you can prove that, and my question is, can you add a DNA repair inhibitor like olaparib, tryptophan, peposertib, to kind of like still enhance the efficacy of temozolomide with a lower dose, but they still have keep the potency of temozolomide? Does that make sense?
Definitely. Actually, this is one of the combinations that we are thinking about by adding O6VG, which is an direct inhibitor of MGMT, to increase the efficacy of temozolomide. That's one of the strategies that we're currently testing in combination. But yeah, definitely because for temozolomide in repair system for temozolomide effect, there are several layers, right? We have MGMT, we have base excision repair, we have MMR system, and for example, as you said, we can use PARP inhibitors to target BER, base excision repair, and improve temozolomide efficacy. But the strategy that we are, as I said, currently testing is combination of O6VG. It's a direct pseudo-substrate for MGMT, to use that to improve temozolomide effect.
Great. I had a follow-up question. Heidi, do you know anything about how the DNA damage repair inhibitors like PARP inhibitors are tolerated in patients with neuroendocrine tumors?
Yeah. So that's the reason why I'm asking, because I mean, we do have a lot of experience with that and clinically. So because we do have the temozolomide olaparib study for FIOPARA, not necessarily for neuroendocrine tumors, and we also have the PRT in combination with olaparib as well. And the whole idea is that we know that temozolomide being an alkylating agent, it causes DNA breaks. And sometimes what happens is through the mechanisms that, you know, was discussed earlier, sometimes they develop drug resistant. And sometimes we can see that, you know, maybe the temozolomide may not be effective. And then that's the reason why adding a DNA repair inhibitors, which it could be, like I say, olaparib, temozolomide, olaparib, peposertib, or tryptophan, that can enhance efficacy. We have seen it in the lab. Clinically is well tolerated, but we need to use lower dose of temozolomide in combination with olaparib, because we know that the two of them can cause cytopenias. But at a lower dose of temozolomide, is fairly well tolerated, and we don't see that many cytopenias. But that's clinically.
That's excellent. Yeah. Thank you.
Can I have a question? Is that okay? All right. Yeah. So we know that temozolomide is very dependent dose and schedule dependent, mainly dose dependent. So when you combine temozolomide and olaparib, was that olaparib that you're using in the clinic?
Yeah. Yes.
When you combine these two, to be tolerable, you have to reduce temozolomide effect. Temozolomide dose. Do you still see the same effect with temozolomide when you reduce the dose? Because that's a question I always have. When you reduce TMZ, it seems that it's not as efficient as the full dose.
Right. Yes. I mean, again, this is clinically, and it's hard for me to say because we don't have, we haven't even get to the number of patients to do an interim analysis. But that said, based on personal experience, not necessarily the data of the study, I do feel that it works well. In an animal studies, a combination of the two have greater cytotoxic activity compared to temozolomide alone.
Very good. Thank you.
Yeah. We have that proof. I mean, if you want, this is a paper that was published in Fiopara. We can certainly share that information with you.
Yeah, thank you so much.
Mm-hmm. Thank you. And I'm sorry, I look forward to the whole talk once it's available on the NETChatter Foundation website. My apologies, I came a little late. I'm sure it was a fantastic talk.
No, I have also another comment about the cell lines, because we found we never used the overexpression system, but we found that in some neuroendocrine cancer cell lines, I think QGP and the thyroid MZ have pretty high SSTR2 expression. So maybe, next step will be to try on endogenous SSTR2 expression as a target instead of overexpression, because we have experience with the other membrane receptor for thyroid cancer, GSHR receptor, and we noticed that there's a huge difference when we have overexpression. Targeting is wonderful comparing to endogenously expressed. So what I remember, and I think we published, Rachel, if you remember too, I think QGPs have highest expression from neuroendocrine tumor, and also MZ, so this is medullary thyroid cancer. They can be used as an endogenous SSTR2 expression.
Yes, that's correct.
It's a great point, because we know that in first of all, HCTs are not neuroendocrine tumors, they're colon cancer. But I actually, for the efficacy, like DNA damaging analysis, we use IMR32 cells that they have endogenous expression of SSTR2. But mostly for imaging studies, we started with these high expressors, HCT, the pair of HCT wild type SSTR2, and then we repeated the study to make sure that in an endogenously SSTR2 expression tumor model, we still can see that signal. We developed that model with H69 that again endogenously express SSTR2. That's a good point, because when we have overexpression, we don't know how much the results are actually close to the reality.
Probably it will be, I'm assuming it will be like almost 40-fold difference between endogenous overexpression and the plasmid.
Yeah, this is a great discussion. We've got about 4 more minutes on the clock. Does anyone have any more questions before we wrap up?
I just want to say thank you for sharing all this, and everybody for pitching in and really asking like really cutting edge questions. And I'm so amazed. It's like I can just sit back for now. Like we've come such a long way, and now that we have these tools to start really like digging into the hard question and the quantitative questions and all that, and it's just it's just overwhelmingly beautiful to see this. So I just wanted to add that. Thank you. Thank you so much for accepting to present, and thank you everybody for really for those wonderful questions and also guiding points that can help this study and other studies to kind of move along, because we're in a really hard field, and it's been really challenging, but it's really nice, and it's really awesome to see how the work is progressing at such a great in this great momentum here. So it's wonderful. Thank you.
Definitely. Thank you for giving me your time to present and for all the great questions.
And something I would like to say, I mean, DNA repair, pathophysiology, and evasion is something that, our branches focus on, and we'll be happy to collaborate with you and anything that you may need to, because we do have organoids, and we can certainly help with some of the information as well as share some information with you, because that's some of the in the lab as well.
Perfect. Thank you so much. Of course. That'd be great.
Yes,
Yes,
thank
we
you.
We'll be also happy to help. We have a lot of SSTR2 model, and we even know how to induce SSTR2 in patients who do not have SSTR2 expression, because this is also a group of patients.
Yeah. So are you using this HDAC Wnt inhibitor?
And we have also another ideas. So yeah, let us know if you want also to include SSTR2 negative patients, because I think this would be quite interesting.
Amazing. Yeah. Perfect. Thank you so much. Well, I'm surprised with all this opportunity. Thank you for the great discussion.
Yes, thank you, everyone. And I completely agree with Poe. This has been absolutely wonderful. And thank you to our speaker, Dr. Solmaz, for presenting and sparking, this wonderful conversation. This is the conversation that we need to have to keep this field going. So it's been an honor to have you speak today. So I'm going to go ahead and wrap up the talk. Stay in touch for information about October, November 10th is the International Neuroendocrine Tumor Day. So I'm thinking we're going to do a special event for that. So follow us on Twitter and stay updated, and I'll let everyone know. So thank you again, everyone, for attending. And this recording will go up on YouTube as soon as I can get it there.
Thank you.
Thank you.
Thank you.
The